That was good.
Great.
And we can hear you. So please, so Jack's going to tell us about cell type specific co-expression network analysis and single nuclei RNA-Seq heart tissue data from inbred HRDP strains. Thanks, Jack.
Thank you. And thanks, everyone. My name is Jack. I'm a research associate at the University of Colorado Andrews Medical Campus. I'm in Laura Saba's lab, and this work was done in collaboration with Caitlin O'Meara and Michaela Patterson at MTW. So I'm a statistician, so I'm working on statistical methods as it relates to this project, but I'll give some overview of the general project motivation before I get into our method. The long-term project goal is to identify genes that are linked to resilience to cardiac injury and tissue regeneration, so understanding the etiology of heart repair, heart conditions using model organisms. The goal of our particular analysis is we have single nuclei RNA sequencing data from heart tissue from 3 inbred strains, 2 duplicates per strain, so 6 total animals from the hybrid rat diversity panel. And we are seeking to identify cell type specific co-expression networks using this data. I'll talk about what all this means in the subsequent slides. Our hypothesis is that these cell type specific networks will provide insight about the genes and cell types relevant to resilience to cardiac injury and tissue regenerationase. So I imagine this is probably review for many in the audience, but I'll discuss it briefly just in case. We're using gene co-expression networks as our way of modeling the co-expression of genes in this sample. The basic idea here is that if the magnitude of RNA expression of 2 transcripts correlates over multiple quote-unquote environments, then the 2 transcripts are involved in similar biological processes. In our case, environments are different cells. So as we see in this plot here, this line plot here, gene A and gene B in blue and red have similar expression, in this case, across these 6 different rat genomes, whereas gene C does not. This means that gene A and B are likely co-functional, whereas gene C is likely not co-functional with either A or B. And we want a way of mathematically formalizing this and then estimating modules or groups of co-expressed genes that we presume are likely to be functionally related. And we're going to do this by first encoding this co-expression as a network using the mathematical concept of a network, which allows us to define a unit of distance, which allows us to do clustering, and that clustering gives us our modules. So WGCNA, whole genome co-expression network analysis, is a canonical method for estimating networks and doing clustering and module estimation on bulk RNA sequencing data, where obviously the RNA sequencing data comes from a amalgamation of cells of different type from a tissue. In single cell data, the sparsity of gene expression within a single cell makes measurement of co-expression difficult. So many genes of interest may not be expressed in a single cell because it's a single cell data and not bulk data, right? And the basic unit of measuring the relationship of 2 genes is Pearson correlation, which is then transformed to estimate the network. And Pearson correlation does not have good mathematical properties when the data is sparse, when maybe 95% of cells do not express a given gene. So we need an adjustment to facilitate network estimation in single cell data, which is the high dimensional WGCNA or the HD WGCNA. What this method does is it first forms so-called metacells, which aggregate expression across groups of similar cells before performing the network analysis, and then kind of proceeds as in classical WGCNA. Co-expression can be measured between metacells from a specific cell type cluster in the RNA sequencing, which allows for estimation of cell type specific clusters. And so we'll get into why that's useful in a second. But basically, when we do the network analysis, it is useful to only use cells of a single cell type. If you use all cells, where cells are of different types, but all cells in the sample, the kind of co-expression information may be picking up kind of general patterns among cells of a single type rather than truly co-functional modules. So it's often recommended to do this network estimation in cells of a single type, which is what we do. So to address the issues with sparse gene expression, cells are grouped into units of similar cells called metacells before analysis. How this is done is a k-nearest neighbor is applied with bagging to construct metacells. These parameters are malleable. They can be chosen, but we chose 25 and 15 kind of per package recommendations in our data. So metacells aggregate 25 cells and are permitted to have 15 cell overlap with other metacells. So metacells are not independent. They don't contain disjoint sets of cells. In fact, there's actually a lot of overlap, up to 15 cells. So these metacells might be correlated a little bit more than typical kind of subjects in a classical WGCNA analysis, but nevertheless, this works fairly well. They're roughly 15% as many metacells as cells when we do this k-nearest neighbor with these parameters.
Jack, let me just put you on hold for a few minutes because we have some special guests. The good news for you, Jack, is that lunch doesn't arrive here in Memphis until 12:30. So as far as I'm concerned, you can chat for quite a while.
Okay, so I'll be on hold.
No, please stay on because I just wanted to introduce Chancellor of the University of Tennessee Health Science Center, Peter Buckley, originally from Cork, England. Good Lord, don't say that. From Cork, Ireland, and been our Chancellor now.
Good morning. Good morning, Jack. I apologize, but the good news is I'm not able to give your presentation, so you will get back to it. I'm delighted you're here. I'm sorry I didn't get to welcome you until the final day. I did have a look at the itinerary. It looks like you've had some really interesting sessions. I particularly would have loved to have been for yesterday's omics session. And then also I'm glad that you're on by hybrid. I think this is the way of the world. And so not only are you talking about cutting edge science, but you're also delivering your message and your dialogue in a contemporary manner. We feel very privileged that you're here. We're very proud of our genetics department and the work that is done here. And our colleagues look forward to learning from you as well as to advancing the field. And now I will get out of your way and go back to Jack. I just wanted to out of respect, welcome you. Thank you all very much. Thank you, Rob.
I just want to point out that Peter has been a practicing psychiatrist for many years and now a senior administrator, but he's still quite productive. So what's of interest to our community, he's been very interested in the interaction between inflammaging or the inflammatory tone in schizophrenia, which is a very hot topic. He had a paper February of this year on this issue where they tested it in a fairly sizable cohort, didn't find anything, which doubly impresses me that these important negative results get published. But when he came to us, he was not only a candidate for the chancellery, but he also was the co-author on 2 Nature papers that were back to back. So he doesn't take much credit, as you can tell, but thank you, Peter.
Jack, can you share your screen again, please?
Yes. Okay, I'll start again. I didn't hear a yes or no, but I'm going to start presenting again. So the data processing for our specific sample is we had almost 36,000 cells obtained across 6 samples from 3 inbred HRDP strains. That's the BNLX, F344, and M250. We did quality control, remove cells with less than about 500 genes, as those may be dead or dying or non-functional cells. Cells with more than 7,500 different unique genes, as these may be doublets or triplets. Cells with greater than 5% mitochondrial genes, or with over 10,000 unique molecular identifiers. After this screening, we had around 29,000 cells. We limited the analysis to only coding genes. This is a classic step taken for WGCNA analysis, because coding genes better resemble kind of a scale-free network, which facilitates the good properties of WGCNA than it does if we use all genes, including non-coding. So we limited the analysis to only coding genes. The median number of unique genes expressed per cell is 1,024, and the median UMI, number of unique molecular identifiers, is 1,690. To determine cell type, we first did clustering based on the UMAP reduced dimensional representation of our data, which looked like this. UMAP clustering had 10 different clusters, as we see the first and second UMAP features plotted here. The three largest clusters are clusters 0, 1, and 2, which we determined to be cardiomyocytes, endothelial cells, and fibroblasts. So how this was done, for each of these three clusters, we looked at differentially expressed genes that are genes that were highly expressed in that cluster and not as highly expressed in the remainder of the data. And by reviewing these sets of differentially expressed genes in each cluster, we're able to assign a nominal or apparent cell type to each cluster, those being cardiomyocytes, fibroblasts, and endothelial. 5,800 cells, 844 metacells were identified as fibroblasts, and around 8,000 cells and 1,158 metacells were identified as endothelial, whereas 8,135 cells and 1,243 metacells were identified as cardiomyocytes. We then use these metacells to estimate the networks and estimate modules. So we see here is a dendrogram of the clustering for each of the three cell types. Again, we did cell type specific network analysis and clustering. So after we derived those clusters, we took only cells in that cluster, only metacells in that cluster to do the network analysis. So as you see, there are 5 different clusters for cardiomyocytes, 5 for the endothelial cells, and 4 for the fibroblasts. Next, we assessed module preservation. Module preservation is the concept of seeing if a nominal module in one data set is preserved or conserved or appears to exist as well in a second data set. So you have a reference data set where you have module definitions, and then a target data set with the same genes where you're going to test if that module is preserved. Notably, this approach does not require or leverage estimated modules in the target data. So you're not kind of like pairing up modules from the target data set and the reference data set. It just uses the network representation of the target data set without actual module assignment. So we assess module preservation using all possible combinations of reference and target data sets. So we use cardiomyocytes as the reference and then see if it's conserved in both the endothelial data and the fibroblast data. Likewise for the endothelial against fibroblasts and cardiomyocytes, et cetera. So every cell type, the modules for that cell type are used as the reference data twice and the target data set 4 times for all of those pairwise combinations. Particularly, we use the Z-summary module preservation statistic, which agglomerates density-based, connectivity-based, and separability-based statistics to get a general summary of how well preserved a module is. We also assess module quality using the Z-summary metric as well, which considers module homogeneity and separability just in the reference data. So it kind of sees how cohesive the module is. The summary statistic, the module quality Z-summary statistic measures how kind of separable the module is in the reference data, whereas the module preservation statistic sees how conserved the module is in the target data. And so now we show the results for all 3 module types. For the cardiomyocytes, we see that all modules are of relatively high quality. As long as the quality is bigger than kind of 2 or so, we say this is reasonable. And then the next 2 plots are the cardiomyocyte and endothelial, right, with the cardiomyocyte modules projected or tested for preservation in the endothelial data, and likewise for the cardiomyocyte into the fibroblast data. These gray regions here, less than 2 indicates basically no evidence of preservation. Between 2 and 10, the light gray area indicates some or moderate evidence of module preservation, whereas the white area indicates a very high degree of module preservation. What we're trying to do here is we're kind of trying to parse out the etiology or functioning of the different modules that we estimate. So the modules are estimated right in that specific cell type only. And it's likely that some of these modules are probably represent cells that serve a very general or central function to cell functioning in general. So these are probably groups of genes that co-function in all cell types. And if we have a module that is well preserved, we believe this is probably representing a general functionality of cells and not a specific pathway to that cell type. So it might be of less interest if a module is well preserved because it's kind of a general pathway and not a pathway specific to cells that we're interested in. And if a module is not preserved, right, like C5 we see here is in the dark gray region, it might be that that module represents a functionality specific to cells of that type. So this is kind of how we're trying to take the module pardon me, the network analysis and module analysis in single cell data and understand whether the modules we find are specific to our cells of interest or more general. And not that one is necessarily of more interest than the other, but it is useful for kind of further parsing the etiology of these cells. This is a behavior that we're going to see relatively commonly as we look at the other two enrichment analysis results as well. That the larger modules, there tends to be one module that's very well preserved. In this case, C1, cardiomyocyte module 1, is very well preserved in both endothelial and fibroblasts, whereas C5, the smallest module, is not preserved at all in endothelial or fibroblast cells. So C1 might represent a group of cells that a group of genes that serve a very basic cell type function, whereas C5 might represent a group of genes that are specific to cardiomyocytes. We see similar results for endothelial. Again, we have this larger module, E2, the second largest module in the endothelial cells. This module is very well preserved. And again, the smaller modules appear to be not as well preserved. In particular, the smallest module, E5, looks like it's not preserved in cardiomyocytes, but potentially preserved in fibroblasts. Lastly, for fibroblasts, now we have only 4 modules. And again, a larger module, in this case, the second largest module, F2, is very well preserved. The other modules are not as well preserved. F3 looks like it is well preserved in endothelial cells, but not well preserved in cardiomyocytes. So that's our intuition, right, is that module preservation may represent to some extent what degree a module is specific to cells of that type versus more general functioning of a cell. So to follow up this intuition, we did an enrichment analysis. We tested each module for enrichment. We took the top 50 module genes of each module and tested them for enrichment against the Descartes Cell Types and Tissue 2021 database. We used correlation with the module eigengene to rank the genes in our module. So KME, which is correlation with the module eigengene, is the correlation between a gene's expression and the first principal component of genes in that module. It's believed that if a gene is well correlated with the module eigengene, it kind of is a central or hub gene for that module. Then we reported all enrichment associations with significant p-values, Benjamini-Hochberg adjusted with p less than 0.1. And for now, we just looked at the most preserved and least preserved module for each cell type. As we see here for cardiomyocytes, C1 is the most preserved module. This is not enriched for cells of any specific tissue type against the Descartes database, whereas the least preserved cardiomyocyte module, that is C5, it is enriched for 4 cell types, 1 smooth muscle, 2 skeletal muscle, and 1 megakaryocyte. So these are tissue types that might be related to cardiomyocytes. This is some evidence, right, that this non-preserved module might be specific to cardiomyocyte cell functioning. For endothelial, the most enriched module, E2, was actually enriched for 2 vascular endothelial cell types. So that's not exactly what we would expect. You might expect it represents, a non-cell type specific module, but in this case, it looks like it does retain some information specific to endothelial cells. Whereas the least preserved module, E5, the smallest one, that was enriched for 4 vascular endothelial cell types. So it looks like that is more enriched for endothelial cells or endothelial cell specific functioning. For fibroblasts, we didn't see a strong signal. Either the most preserved or the least preserved module is enriched for cells of any type. Interestingly, only 1 fibroblast module was enriched. That was F3, which is highly enriched for vascular endothelial cell types with 16 significant associations. If you look at the F3 here, you see that F3 is not enriched for cardiomyocytes, whereas it is enriched for endothelial cells. So it looks like our enrichment analysis is picking up that signal as well. That's a conclusion. This is kind of a first step or a beginning of an analysis to understand how to isolate modules that are specific to cell type functioning using single cell RNA sequencing data. And thanks to the co-authors and our support. And with that, I can take any questions.